terms related to this DNA and RNA. The first term is replication. It simply means DNA making another copy of the DNA. Replication means duplication. The second term is transcription. As I said earlier, DNA also is responsible for making RNA, and the process is called as transcription. Right. Next, translation. Now, the DNA which has made RNA, now when the RNA which is responsible for making the specific protein, it is called as translation. And one more important term is reverse transcription. It is a reverse of transcription. So transcription is DNA making RNA, and reverse transcription will be RNA making DNA, and that is reverse transcription. So these are important terms related. So we will understand these processes one by one in the coming part. The first part we're going to start with is the replication. As you can see in the screen, a parent DNA is replicating to make daughter DNA. The two daughter DNAs are the exact replica of the DNA. They have the same quality and same quantity of the DNA. But in that, you also notice that in the daughter DNAs, both the strands, in the daughter DNA, both the strands are new, yeah, both are old, yeah, one is old, one is new. And yes, because you can see that in the daughter DNAs, one strand is old, the other is new. It means that the replication of DNA is semi-conservative. So semi-conservative means one strand of the parent DNA is always conserved in the daughter DNA. Hence, the process of replication in eukaryotes is semi-conservative. So let's understand the semi-conservative method of self-replication of DNA in eukaryotes. Now, before we start, let's understand the basic requirements of replication. Now, replication requires mainly three types of basic requirements. The first type are the substrates. As you know, DNA is made up of nucleotides. So if DNA is made up of nucleotides, so new DNA synthesis will require new nucleotides. Then DNA, definitely the DNA itself, which is to be replicated, we call it as template, has to be a basic requirement. And the most important requirement is the enzymes and proteins which are going to bring upon this process of replication. So enzymes or proteins related to the replication of DNA are a most important basic requirement of replication. Now, first is the substrates. Our substrates, they should be nucleotides. But during the replication, the nucleotides are supposed to be activated ones, because during replication, when they are going to be a part of the new DNA strand, that new DNA strand synthesis is going to consume some energy, and these nucleotides already are to be activated. So in the activated form, they are not monophosphates. As you can see, they are actually triphosphates. So instead of deoxyadenosine monophosphates, the activated nucleotides here are deoxyadenosine triphosphates and the other so on. So these are the substrates basic requirement. The template, is the original DNA which is to be replicated is called as the template, and definitely the enzymes and the proteins also. Now one look at how the activation of nucleotides occurs. Now this is a mononucleotide which needs ATP for its activation, and you will note that how the ATP is giving its two phosphates bonds to the mononucleotide and making it into a triphosphate nucleotide, which is an activated nucleotide. For example, DAMP, that is deoxyadenosine monophosphate, has been converted to deoxyadenosine triphosphate. So that's how the activation of nucleotides, which already happens in the G1 phase during the cell cycle, and then they are ready for the replication. Move on to enzymes and proteins. Now, enzymes and proteins are not one or, but there are many of them. Now, we have classified the enzymes related to replication into two categories. One category is the key enzymes, the most important. The other category is the other enzymes, which also help the DNA in the replication. Now, key enzymes include four of them. The first is the helicase. Now, in order to replicate, the DNA is tightly coiled around itself. Now, if it has to replicate, the coils, the helix has to actually open up. So to open the helix, the bonds in between the two strands have to break, and then the helix should open. So to break the hydrogen bonds and open the helix, there is this enzyme responsible which unwinds DNA. It's called as helicase. The second enzyme is the most important one, the DNA polymerase, responsible for making the new strands, which joins the specific nucleotides to a specific template, is very, very important. DNA polymerase. It elongates the DNA chain with specific nucleotides. The third important key enzyme is primase. Now, in order to start a new strand, the first nucleotide has to have one hydroxyl group to bond with, and that hydroxyl group to bond with is provided by special nucleotides, RNA nucleotides, which is called as RNA primer. Now, it is the enzyme primase which synthesizes this RNA primer, which helps in to initiate the new strand of DNA. We will see in the process ahead. And the last key enzyme is the ligase, which actually joins two DNA strands. So two fragments of DNA can be joined to each other. We will see in the next part, there is going to be something called as Okazaki fragments, which will be joined using this ligase enzymes. Move on to the other enzymes. Now, other enzymes again are grouped into four. The first one is SIP, that is specific initiator protein. Now, it is the protein which decides where the replication should start. Now, the points where the replication starts is called as origin. There is a short sequence there where the replication is going to start. The sequence is also called as ORI. ORI sequence is responsible to begin the replication. The second key enzyme, the second other enzyme what we have is SSBP, that is single-stranded binding protein. Now, this protein is important because once the helix is open, once the double-stranded DNA after opening has become single-stranded, we have to make sure that they do not again make a helix. So SSBP binds to the single strands and stabilizes them so that they don't make an helix again. The third enzyme is topoisomerase. Whenever any tightly coiled structures you try to unwind them, they will always have the torsion around them of coiling. So in here also, when the DNA is uncoiled, the torsion relief has to be there. So topoisomerase will release the torsion. The coiling tension will be relieved by topoisomerase. It is very important again. And the last enzyme here, which is going to be involved, is TER binding protein, which prevents the helicase from further unwinding and facilitates termination. So whenever the helix is going to unwind the helix, there has to be some break to it, some stop to it. So this protein is going to help it to prevent further and unnecessary opening of the helix. So these are the all types of enzymes which we will see in the process, which are going to help in self-replication of the DNA. Starting with, as I said, there is an SIP which decides from where the replication should start. Now in eukaryotes, in the nucleus, the DNA is very long. Now such a long DNA is going to take a lot of time to replicate if it starts at only one point. So it's not one, but several such points are called as origin, where the replication is going to start simultaneously, which is going to make the process a little more faster. So the name of these regions is called as origin, and the sequences are called as ORI sequences. Now, once the ORI is decided, there is an enzyme which is going to cut one of the strands. The enzyme actually is called as an endonuclease enzyme, which cuts one of the strands so the helix can open and the helicase can basically start unwinding it. So the cutting of one of the strand is called as incision. Once insertion occurs, the helicase will break the hydrogen bonds between the two strands and help in opening and unwinding of the helix. Now, after unwinding of the helix, the DNA will appear in this Y-shape arrangement. Now, this Y-shape arrangement also has a name. It is called as replication fork. Now, mind you, students, here this is only one side of the DNA. Don't forget, wherever the replication fork is formed, there is going to be one more fork opposite to that in this manner. And such forks, when they face each other after unwinding, the arrangement will be now called as a replication bubble. Now we are focusing on one of the uh, helix, one of the fork which has opened because of the helicase enzymes. But yeah, here you also remember that replication is going to start not in one, but here both the directions are going to do replication. So DNA going replication is bidirectional also. And if the replication starts at the end or the beginning of the DNA, it is going to be only unidirectional. So remember, replication is unidirectional or it can be bidirectional. If it starts somewhere in between, it is bidirectional. If it starts at the ends, it is going to be unidirectional. Let's focus on one of the Y replication fork. Now, as I said earlier, the key in the other enzymes, there was an enzyme called as a protein called as SSBP, single stranded binding protein, which is necessary to bind to the single strands so that they do not form the bonds again. So SSBP will help in that. And as I said, when the DNA is going to open up, when the helix is going to open up, now beyond that, beside that area, the DNA which is coiled earlier is going to have lot of torsion around it. So that torsion strain in the helix has to be relieved. And remember, what enzyme is going to help there is topoisomerase. So topoisomerase enzyme will come there, will break some bonds of the DNA there, and will release the tension and will rejoin that part again, and that's it. So once that is over, now the replication is going to start. Now, before we start, we also should know that the two strands of the replication fork are basically the original strands. They are called as templates. Now, both are templates, but remember, both the templates don't start replication simultaneously. The replication first starts in the leading template, which is seen in the direction of 5' to 3', and the lagging template, which is visible in the direction from 5' to 3', but in the opposite direction. So leading strand, as the name says, is going to replicate first. And do you remember, to start the replication, they require one hydroxyl group to join the first nucleotide. So there is going to be an enzyme called as primase, which is going to allow the attachment of RNA primer. So RNA primer, which is made up of around 5 to 10 nucleotides only, they're short RNA nucleotide sequences. The purpose is to provide the hydroxyl group to the first nucleotide which is joined. Now, once the primase functions, it attaches the RNA primer. Now comes the most important enzyme, which is going to attach specific nucleotides on the new leading strand. Now, remember, this replication also has a pace. The replication occurs at attachment of 50 base pairs per second. And DNA polymerase is going to attach specific nucleotides which are already present in the nucleus on the leading strand. They are going to join the nucleotide by making the phosphodiester bonds. Specific nucleotides will be joined and the new continuous leading strand is formed. And the ends of the new strand, 5' end and 3' ends. This is the leading strand which is made continuously. So as you understood that this DNA polymerase always synthesizes the new strand in the direction of 3' to 5' on the template. But remember, when both the strands of the DNA should be parallel, the replication of both the strands also is going to be parallel but in opposite direction. So polymerase keeps on following the helicase in the direction and makes a continuous strand. But the lagging strand, that is the opposite strand, is going to also make, but the direction of the new strand is not going to be the direction in the helicase, but it is going to be in the opposite to the helicase. That is going to be the lagging strand direction. Let's understand the formation of lagging strand. Now students, remember, the lagging strand does not form continuously because now it is going to start from the helicase towards the end, towards the 5' end of the template, on the lagging template. Here RNA primer hence is going to be formed near the helix where it is opened. RNA primer will come as usual. DNA polymerase will make the new strand as expected, but the direction is in the opposite side of the leading strand. So once that is ready, now this is not going to be so continuous. Now this strand is going to be very short. Why? Now why? Because now this DNA polymerase, once it has made the lagging strand, the continuation is not possible because the next Y-shaped replicating fork has already finished with its replication. Remember that bubble which I had shown had already finished its replication on the opposite side's leading strand. Now the new fragment will be formed once the helix opens the helix further. The helicase will open the helix more and more ahead in the given direction, and then it is going to allow the further replication of lagging strand. That means lagging is going to have breaks, and these small fragments are having a name. They are called as Okazaki fragments. So around 100 to 200 nucleotides make one fragment. So at a time only one fragment is made, but every time whenever there is fragment is made, a new RNA primer is necessary. Like that, multiple fragments will be there and then they will make the lagging strand, the new lagging strand. But the problem is again the RNA primers. Now some enzyme has to come and remove these primers and replace them with the DNA nucleotides. Because these primers have RNA nucleotides, they are to be replaced with the DNA nucleotides. So again, another type of DNA polymerase will come and replace these primers. Here it comes and is going to replace them. And then after replacement, you can see again there is a small gap. Now, again, the gap needs to be filled by bonds between the two nucleotides, and there is one enzyme who's going to make this possible, which is going to make to join all these Okazaki fragments together, and name of the enzyme to fill this gap and make this bond is DNA ligase. Ligase is going to join all these Okazaki fragments by making the phosphodiester bonds and ultimately making the lagging strand also continuous at the end. So thus, this was the formation of the lagging strand. So now when both the strands are ready, the two daughter DNAs are ready. So this is how both the templates have made their leading strand as well as lagging strand. But remember, one strand leading was continuous and the other lagging strand was discontinuous because it made those fragments called as Okazaki fragments. Later on, ligase rejoins them and makes it continuous. So a quick recap of all the enzymes which we used in the replication process on the screen. So key enzymes and the other enzymes, I hope you remember, I hope you have understood how these enzymes have worked. Now, is it possible that during replication, there some error could happen? Like during replication, adenine should pair with thymine. But is it possible that the DNA polymerase did a mistake, did an error, and instead of thymine, the adenine has got paired with guanine? Or instead of guanine, one cytosine has come. So such errors, the chances are definitely there. The chances are definitely there, but again, not to worry. There's a mechanism after a replication. It's proofreading and DNA repair. There are specific enzymes who work for this process. Now suppose these two are the parental strands and they are doing their replication. DNA polymerase attaches the specific nucleotides to on the specific pairing, making it a new leading and lagging strands. Now, during that process, you see, did you notice there is one error made by the enzyme? Now, in opposite to cytosine nucleotide, they have attached the thymine nucleotide, which is an error. Now, what should happen? There is this enzyme which are able to recognize the errors and make corrections into them. And it's corrections के लिए again there is the same enzyme DNA polymerase going to do the necessary correction and it is going to replace the thymine with guanine nucleotide and it is going to be repaired. So this is basically the proofreading and DNA repair. Hence, the chances of mutations are very low because of these proofreadings and DNA repairs. So this was the process of DNA replication with the specific enzymes. Now, these are some differences between prokaryotic and eukaryotic DNA replications. You can see the primer lengths of the prokaryotes and eukaryotes are different. The primer lengths in prokaryotes is quite 50 nucleotides approximately, and eukaryotes have around 9 nucleotides. The DNA polymerase enzymes making various functions are also of 3 types in prokaryotes and 5 types in eukaryotes. Origins also. Prokaryotes having a very small amount of DNA have only one single origin, and eukaryotes having an abundant amount of DNA needs to have multiple origins. And because small amount of DNA, the Okazaki fragments are also not very much higher in number. They're very low in number, but the number of nucleotides in the Okazaki of prokaryotes is around 1000 to 2000. Eukaryotes is around 100 to 200 nucleotides, I told you. The rate of replication is also high in the prokaryotes because their rate of replication high because their multiplication also is very high. So around 500 nucleotides per second in prokaryotes and 50 nucleotides per second in the eukaryotes. Remember, the prokaryotic DNA replication is called as the theta replication. The types of polymerases and their functions. So prokaryotic polymerases are 1, 2, and 3 specified functions and are mentioned in the table on the screen. Eukaryotic polymerase have alpha, beta, gamma, epsilon, and delta. So these performing various functions. One is making Okazaki, the other one is doing proofreading, one is doing DNA repair, the other one is mitochondrial DNA synthesis, one is doing leading and lagging strand. Like that, these are the different functions performed by different types of eukaryotic polymerases.